Good morning. My name is Robert Klein, Chairman of the Governing Board of the California Institute of Regenerative Medicine. We give special thanks to UC San Francisco for hosting us here this morning. Today we are going to focus on the value and critical importance of basic research. I would remind everyone that it was a seed grant that led Katrina Jameson to her breakthroughs for the myeloproliferative blood disease, where she is now in a phase 1 human trial with potential to reduce and or eliminate a tremendous amount of human suffering. Actually, removing patients from bone marrow transplant lists in what they thought was an endgame of life. This morning, I will have the privilege of introducing Dr. Hawgood, who is a very distinguished member of our board, and who has now been named both Dean, subject to the Regent's concurrence, of the School of Medicine, and Vice Chancellor for Medical Affairs. It is absolutely vital that basic research continue in this country, and with the support of visionary, insightful leadership like Dean Sam Hawgood's, we have tremendous opportunity to move forward with the basic discoveries that will be so critical to translational medicine and the challenges before us. The Dean is the former chief of neonatology and an expert in caring for newborns with birth defects. He is a member of the Fetal Treatment Center and Pediatric Heart Center at UC San Francisco, and a professor and a chair of pediatrics, while being an associate director of the Cardiovascular Research Institute. He is a former president of the Society for Pediatric Research. It is the quality of leadership at this level that provides national and international leadership for UC San Francisco and the California research effort. It is important to realize that he is part of the international delegation of talent. He is on loan to us from Australia, along with our formidable president, Dr. Trounsend. With that, I would like to introduce Dean and Vice Chancellor, Dr. Sam Hawgood.
Thank you very much for that warm welcome, Bob. And it's my pleasure to be able to host this meeting here at UCSF, our Mission Bay campus, and I welcome you all here. For those who aren't completely familiar with UCSF, Mission Bay is the newest of what are 6 major sites in the city for the University of California, San Francisco. And just mention briefly what we're trying to achieve here, because I think it's relevant to the spotlight that we have this morning. Just 8 years ago, this was a brown field. It was an ex-rail yard. There was nothing here. We have built a research campus that started as an expansion of our basic sciences, and obviously that's what we're going to talk about here this morning. Became a home for expansion of our structural biologists, our computational biologists, and a place for us to expand into some of the new areas of basic science, systems biology, synthetic biology, etc. They're mostly the buildings that you see right now, but we're in phase 2 of construction now. We have just opened a cancer research building, and early next year we'll open a cardiovascular research building, and we'll begin construction on a neuroscience building. So that's really phase 2, where we're bringing translational research to bear. And then within 3 years, we intend to break ground on a 300-bed hospital just immediately to the south, which will bring clinics and patients here so that we can mix the basic science, translational science, and clinical care. The unique part of this campus and what gets our juices flowing is within a 5-minute walk of this building, there are at least 6 major biotechnology companies that have migrated here because of the research campus that we're building, and there are plans for at least another 6 or so to join us in the next 2 to 3 years. So that's really the fourth leg to the stool here as to how we move things to the clinic. The progress that CIRM has made in the last few years, I think, is nothing short of stunning. Since joining the board, I have been absolutely amazed at the speed at which scientific progress is occurring in the field of stem cells, and the excitement that is building as the disease team grants are coming online, and we are clearly moving towards clinical application. But I think it's critical that we remember basic sciences, and I'm particularly pleased that the spotlight this morning is on basic sciences. And I'd like to just briefly go back a little over 60 years ago with a quote from Dr. Vanderveer Bush, who at that time was the Office of Scientific Research in President Roosevelt's administration as the Second World War was winding down. And he was charged with developing a template for the federal government in thinking about how the rapid progress of science during a period of war could be used to fuel the American economy and not to lose the impetus that the scientific progress during the war had made. And in his report to Roosevelt, which was really a remarkable document even 45 years later, he said that new products and processes are not born fully grown. They are founded on new principles and new conceptions, which in turn result from basic scientific research. Basic scientific research is our scientific capital. And I was particularly excited and enormously encouraged that President Barack Obama took that last sentence, basic scientific research is our scientific capital, and used it in his first address to the NIH earlier this spring. In that same address, he obviously indicated his strong support for stem cell research. So while I'm incredibly excited about the progress we are making towards translational and clinical application of stem cell sciences, I'm also very excited that the scientific staff of CIRM and the board have taken a very balanced approach, and we are continuing to invest in basic science. Clearly, the individuals who started investigating how viruses infect bacteria did not know at that time that that ultimately would lead to recombinant insulin and, of course, an industry we now call biotechnology. Just 18 months ago, at least, the word IPS, or induced pluripotential cells, was not in my lexicon. I'm sure it was in some of the others who are more deeply involved in this area of science. But it came about because of an attempt to understand the basic reprogramming that goes on to achieve pluripotentiality, and now we're talking about the potential clinical application of these cells. So time is being accelerated, but basic science is still absolutely needed.